ಒಂದು ನಿಮಿಷ ವೇಟ್ ಮಾಡೋಣ. ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ ಲೈವ್ ಹೋಗ್ಬೇಕು. ಜಸ್ಟ್ ಐ ಆಮ್ ವೇಟಿಂಗ್ ದೇರ್. ಅದು ನಂದೇನೋ ಸ್ವಲ್ಪ ಪ್ರಾಬ್ಲಮ್ ಸ್ಟಾರ್ಟ್ ಆಯ್ತು, ಸೊ ಅದಕ್ಕೆ ಐ ಆಮ್ ಲೈವ್. I start recording, so I think everyone can hear me, right? So we'll start the class without wasting any time, okay? Because only 1 minute is over before we're going to start the all introduction and everything. It will be complete. It will be 5 o'clock, right? So okay, yes, today we are dealing with the two things. What are those things then? The first things we will discuss the variation and DNA and chromosome numbers during the cell cycle. I told you, if you read the mitosis and meiosis, you will get it quickly, okay? No issue. Otherwise, you may need to read 1 or 2 times, okay? But it's easy. I'm telling you, it's easy, dead easy, okay? Once you understand, you will never like forget this one, okay? So second one is like morphology of flowering plant. We will start that particular one, okay? So we'll go through the little bits of the things, okay? So I'm revising the mitosis, meiosis also for a little bit. So for the first thing, variation of DNA and chromosome number during the cell cycle. That means the DNA number and chromosome number, both of them are not constant throughout the cell cycle, okay? It's normally varying, okay? So it's varying from the one particular phase to the another particular phase. It's majorly depend upon the segregation of chromosome and replication of chromosome, everything, okay? Before we're going to see what are those and how it will change, we have to understand the first thing, what are those annotations or how we do the annotation, okay? The first one, 1 second guys, I will share one more time. Same thing, it's like not working properly. Okay, I think you can see this properly now, right? So there's some lagging. I don't know. Sometimes Zoom will happen. But yeah. Good evening, Gopika. Yeah, I'm starting the chat box. Yep. So good evening, guys. First, we will see the annotations. How we will mark that. Okay. Don't confuse in these things. First one is the number of DNA we are referring here. Okay, that means the DNA content. We can't exactly say like how many like different genes are there, because genes are always like abstract term, okay? It's only based on functionality and everything, but you can still say the number of DNA, because how much long the DNA may be, it will segregate into form a single chromatid only, right? It may be so long or several meters long, it will make a single chromatid only. So we normally refer or count the DNA number in the number of chromatids, okay? So you can see this one is having 1 and this one is having 2 chromatids. So DNA number 1 will be 1 and 2 here, okay? How we how we write it? We normally write it with a smaller term called C, okay? It's just a notation for the DNA number C, okay? Don't confuse with the chromosome number, it's N. DNA number will be notation will be the C, okay? You have to remember this particular one. So how can I write the chromatid number or DNA number of this particular chromosome? Here the chromatid is single only. Remember, these are arms. These are not separate chromatids. This complete thing is a chromatid, right? So you can call it as the 1C, okay? As you can see here, this one. So this one I can call it as 2C, right? Because it's having the 2 chromatids. So exactly same thing. When you come to the chromosome number, okay, when you come to the chromosome number, how can you say this? The chromosome number is equal to number of centromeres, okay? We already discussed this also. So chromosome number will be notated with N, okay? So don't confuse with like, don't make it like reverse or anything. Chromosome number is N, DNA number is C, okay? So number of centromere you have to find. Remember, not centrosome or centriole, something else. It's a centromere. Okay. So how many centromere in this one? This is only one centromere. So I can call it as something like 1n, right? 1n, normally notated with small n, right? So what about this one? This one is having 2c number of the DNA. But what about the chromosome number then? Remember, even though it may have a 2 chromatid, the centromere is still one only, a single centromere only joining the 2 chromatids. Still, this is also 1n only. Okay. So based on chromosome number, we normally call it as like haploid, diploid, and triploid. Right. Sorry. I will write down one second. Oh, diploid and triploid. Right. How can I write these diagrams then? Okay. How can I able to write these diagrams? So I don't know why it becomes so slow. Okay. So haploid, I have to write it as the single centromere. I can write like this. This is also haploid. Or I can also write it something like this. Right. This is also haploid. Both of the things are haploid only because only one centromere is here. But what about diploid one? Diploid, how many centromere it should have? It should have 2 centromeres, right? I can write it something like this. As you can see here, 2 centromere are here, right? Because these are homologous spaced. And exactly same thing, I can also write it something like this, right? These things are always homologous. Okay, so for homologous only you will get the diploid. So exactly, what about the triploid then? You already guessed it, right? I can write it something like this. Okay, or I can write it something like this. Okay, so this is the triploid. The chromosome number varies along with the number of centromere, not about the number of chromatids. Number of chromatids may be anything. Okay, it may be 1 chromatid or it may be 2 chromatids. But the DNA number is always depends on the number of chromatids. That's why if it is only 1 chromatid, it will be 1C. If it is more than like 2 chromatid, it will be 2C. Normally only haploid, diploid this single and 2 chromatid condition only seen, not more than that will not normally won't see. Okay, that's why that much is enough. So understood, right? DNA number notated with C, and you have to tell based on the number of chromatids. Chromosome number notated as N, you have to tell based on the number of centromere. Okay. Good evening, guys. So we will move on to the next page then. It's not going. Give me a second. I'm closing everything and I'm like doing it one. Yeah, now it's working properly. No, chart is not working properly. I think today same thing happened with Shiva sir class also, right? So something happened, like something is stucking. He is also saying same thing, like something is not working, everything becomes slow or something, right? Huh. So we'll move on then. So one question came, a simple question. You solve this, then we can move on to the next one. Okay. The question is simple. How many mitotic divisions are needed for a single cell to make 128 cells? Okay. They're asking how many mitotic divisions are needed for a single cell to make the 128 cells. Okay. So option A 28, option B 32, option C 7, and option D 14. I'm launching the poll now. So you can answer in the poll, or who are in the YouTube live chat can answer in the YouTube live chat. Okay. So no issue, take your time and answer. I think it's one of the easiest question, right? Okay. Simple math, yeah. How many mitotic divisions are needed for a single cell to make 128 cells? Okay. So they are asking how many. Okay. So 30 seconds is over. Still you guys have 25 seconds. I'm ending the poll in 15 seconds. You guys still have the 15 seconds. So voting is very low. Okay, I'm ending the poll then. Okay, I'm showing the results. 3% chosen option A, 58% B, 30% C, and 10% D. I know some of you are wanted to answer, but the time is over. I can't do anything, right? So you should be very quick. How many mitotic division? Mitotic division is a repeated kind of the division, right? So they are asking from a single cell. So one cell is there, it will give rise to 2, 2 will give rise to 4, 4 will give rise to 8, 8 will give rise to 16, 16 to 32, 32 to 64, and 64 to the 128. Right, now we will count how many divisions it happened. 1, 2, 3, 4, 5, 6, 7. Absolutely. Okay, simple math, right? The correct answer is option C, 7 divisions. So 7 divisions are needed to create 120 cells out of from the one single cell, right? So so many people are correct. Okay, exactly, correct. Okay. So now another one question. Let me hear this question properly. Majority is in wrong way. So some of you are taught in different way, I think. Okay. So now another one question. Hear me properly. Okay. So same thing. I will change this mitotic into meiotic division. Okay, meiotic division. Then what will be your answer? I'm not launching poll, so you have to answer in the chat box. Okay. How many meiotic divisions are needed for a single cell to make 128 cells? Okay, how many meiotic divisions are needed for a single cell to make 128 cells? Okay, so can anybody answer me? What division 136 are found? Okay, I'm just waiting. Okay, like. Okay. I will tell the answer. Okay. So the issue is literally it's not possible. Okay. It's not a possible process. Why it is not possible then? Okay. So I will write why it is not possible. So meiotic cells, what happens? A single cell will give rise to the 4 cells. Here, if it is a diploid, it will give rise to the haploid cells. That's it. Over. This haploid can't go through the meiotic process one more time, right? This can't go through the meiotic process one more time. So meiotic division is not repetitive divisions. Okay, meiotic divisions, it's not a repetitive division. Once it's done, it will give the haploid cells that much done, right? So you can't create 128 cells from one cell or many cells from a single cell through the meiotic process. Okay. But they still ask a question like this. Okay, they asked a question something like, how many meiotic divisions are needed for creating 100 gametes? Okay, they asked like, how many meiotic divisions are needed to create 100 gametes? Now probably you guys can answer. Okay, they asked how many meiotic divisions are needed to create 100 gametes. Now exactly. Okay, I'm really happy for you guys. Exactly. So for creating 100 gametes, you need how many cells? You need 25 cells, because 25 cells will give rise to the 100 gametes. So 25 cells, 25 diploid cells will give rise to the 100 gametes or the haploid cells. So this 25 diploid cells will go through the 25 meiotic process. That means each cell will go through one meiosis. So in the same way, 25 cells, 25 meiosis, and it will give rise to the 100 cells. Okay. So you have to calculate in that manner, not like mitotic creation. Okay. It's different. I think you understand that, right? So, okay, we'll move on then. So if you guys don't understand, just make it a math for yourself. Okay. Then you will guys start to understand. Yep. I wrote certain things here. I don't know where it went. I think I wrote something. Leave it, no issue. So first what we discussed, we will discuss about the variation of chromosome and DNA content in the mitosis process. Okay, if I note the different phases, this one is G, this one is the S phase, this one is G2 phase. Okay, don't mind about these things. Okay, next one is the prophase. We will leave this one, and this one is a metaphase, and this one is the anaphase, and this one is the telophase. Okay, first one, DNA content we have to find. Okay, DNA content. So how can we write? We can write it like C or N. It is C, right? So DNA content, and we have to also write the N. N means what? The chromosome number. Okay, chromosome number. So now we assume that, okay, now we will assume that it's a diploid cell. Okay, it's a diploid cell. We will assume like that. Okay, so what happens in G1 then? G1, the chromosomes will be something like this. Okay, something like this. It's not yet divided or anything. So it's a diploid, I already told. So you can write it as like 2N. Okay. And how many chromatids are here? One chromatid and two chromatid. You can write it something like this, 2C. Okay. But when it goes to S phase, then what happens? The DNA duplication is happening, right? DNA replication is happened. That's why the chromosome will look something like this. Then how can you write it? Here, the 2C will become how much? The 2C will become 4C, because here only two chromatids are here, but DNA is double now. DNA replication happened now. So what is happening? That will give rise to two chromosomes. So same two chromosome only remaining, but the chromatid number is get doubled. 1, 2, 3, 4. So 4C. Understood? I'm repeating one more time. In the G1 phase, it's a diploid cell having a single chromatids or chromosomes homologous. But during the S phase, during the synthesis phase, for onion, given that N block in cells, I didn't understand that. So what is happening? DNA content is doubling. So it is giving rise to extra chromatid. That's why the DNA content is getting doubled from 2C to 4C. What about chromosome number, guys? What about chromosome number? What about chromosome number in the S phase? Can anybody tell me? So what may be? Exactly. It will be the same. Why it is same? Because here only chromatids are getting doubled, not the centromere. You can see centromeres are only two still here, right? G2, no changes will happen. Same thing. Okay. Here there is no changes happening to the chromosomes. Only condensation is happening. Prophase also same thing happening. No changes. Okay. We are leaving this one. And metaphase is also same thing only. No changes. Okay. The next change will happen in the anaphase. Okay. Anaphase, what is happening here? Anaphase, the chromosomes are getting separated, or chromatids are getting separated. That means centromeres are dividing, right? Now, what may be the DNA content? What may be the DNA content if I take a single half of this cell? If I take the single half of this cell, if I take single half of the cell, I'm not telling total cell, only single half of the cell I'm taking. So this is how, One second. This is our chromosome until here. Okay. This is our chromosome until here. Here what is happening? It is get separated. Only this will go to this side and this will go to this side. Right. It's moving away. This is moving away in anaphase. Just see how many chromatids you can see. You can only see two chromatids in one side, right? A single side you can only see two chromatids. So you can write it as like 2C. Okay. And what about the DNA what about the chromosome number then? The chromosome number is remained same only, even though the changes happened. What happened here again? So even the changes happened, you can still find only two chromosomes. It will still 2N only. So telophase, no changes happening, 2C and 2N only. Okay. So I'm writing it one more time. As you can see here, so in this chromosome or in the anaphase, what is happening? The centromeres are getting doubled. Centromeres are getting doubled. So centromeres are moving in different direction. Okay. The chromatids are moving in different direction. So it's coming to the initial step only, right? Initially, you can see here G1. In G1, as you can see here, only two chromatids are here, homologous chromosome or homologous chromatid, you can say. Exactly that position only it's coming back to, right? As you can see here, only 2C in the initially it's only 2C. It's again coming to the 2C only. And initially it's a 2N and it's again coming to the 2N only. Okay. So mitosis, you may take how many chromosomes or how many different times of chromosome numbers. Ultimately the end point or the daughter cell will have the same DNA content and same chromosome number as like the parent. Okay, so that's why you can call it as like G1 is equal to the telophase or the next G1. Okay, so DNA content and chromosome content, everything is same. That's why you can call this as the equational division. Equational division, because it's maintaining the chromosome number and it's maintaining the DNA content. Okay, understood. So next is the meiosis. So up to that we are not making any changes. Remember, up to the prophase we are not making any changes. So in meiosis also this one is same only. Okay, meiosis, because interface is same in mitosis and meiosis, both the things, right? So just remember about the G2 phase. How many things will be there? It will be telophase happened means cytokinesis is also same only, right? Cytokinesis is only the changes in the cytoplasm, not related to the this one DNA. So there's also same only 2C 2N only. That's why I haven't written these cytokinesis once again, because telophase means cells are getting separated, right? We are only considering one cell. That's it. So try to remember this one, 4C and 2N. I'm writing the same thing in here also. C and N. I think C you can understand DNA content, and N you can understand chromosome content. Okay. Initially, how much is there in the prophase? Exactly like this, 4C and 2N is there. Okay, 4C and 2N. Decondense. Yes, Sachin. When it's go for decondensation, you can't really see the chromosome. That does not mean that the DNA will be like different. Okay, decondensation happened or not, the chromosome number will be there only, like that only, but you can't really see it because it's formed in a chromatin thread. Okay, it will be there, but it's in a chromatin thread. That's it. Okay. So we will move on to here. So what is happening here? 4C and 2N is there. Is anything happens to metaphase? No, nothing happens in metaphase, same thing maintained. But here look, in the anaphase, I can write initial state something like this in the prophase. Okay, so as you can see here, 4 chromatids are here and 2 centromere is here. But as you can see here, in the anaphase, what is happening? One chromosome, complete chromosome is going one side, and another chromosome is going one side. Okay, now what may be the DNA content? Can you guys guess it? What is the DNA content here? DNA content. Okay, in a single cell, what may be the DNA content? Exactly, it will become to 2C. What is the this one chromosome number? What may be the chromosome number? Chromosome number. Exactly. You can write it like 1C, or I can simply write it like, sorry, as 1N, or I can simply write it like N. Both are same only, right? Remember, in the anaphase 1, what is happening? Because of the moment of or segregation of homologous chromosome to the two different poles or two different cells, the chromosome number and DNA content both are getting reduced. Okay. No, it's reduced, Tanmay. Okay, chromosome number not doubling here, but chromosome number is getting reduced here. Okay, chromosome number getting reduced in this one. So this particular one, so here is also same only maintained in telophase or cytokinesis. So that's why our first division or our first meiosis one is called as the reductional division. Okay, reductional division. Why we call it as the reductional division? DIVN means reductional division. Okay. Why? Because initially it's a 4C and 2N chromosome number, but as you can see here, after M1, it is 2C and 1. That means half of the initial chromosome number and half of the initial DNA content. Okay. Then what happens? We will move on. Meiosis is not yet over. Meiosis still have to go, right? So in the prophase, same thing maintains. Same thing, and in the metaphase, same thing maintains, 2C and N. Okay. In the anaphase, look here, again changes happening. Here one homologous chromosome is here, metaphase, like this, I can write it like this. Okay. In here, what is happening? Here is also one more is there. So here, the centromere breaking is happening. That means single chromatids is moving in different direction. That means this will go to this position, this will go to this position, this will go to this, this will go in this direction, and this will go in this direction, right? So centromere breakage or chromatid separation is happening. Guys, can you now tell me what may be the this one chromosome number and DNA content in the anaphase? You just have to write the like things, right? Exactly. Here the DNA content is getting reduced to 2C to 1C. Okay, why it become 1C? Because until this, we have two chromatids, right, in a cell. But here I can see only one chromatid left. That's why we call it as like 1C, one DNA content only left. Okay. What about the chromosome number then? The chromosome number here is also same, only one centromere. Here is also only one centromere. That means no change. That's equal to 1N only, or I can simply write it as like N. Okay. So what happened here? I'm just making it a square. Okay. As you can see here, in this phase to this particular phase, in this particular phase to this particular phase, if you see, if you see the chromosome number, it's almost like exactly the chromosome number will be same only, right? The chromosome number is not at all changing. The chromosome number not at all changing. It's same. That's why we call M2 or M2 as the, I will write it above only. Okay. M2 is equational division. Equational division. We call it as like equational division. Now you guys ask me, so really like here from 4C to 2C is happening, and from here to 2C to 1C is happening, but we called it a reductional phase. But why we are calling this one as the equational phase, right? Because we are considering the names, we majorly consider the names based on the number of chromosomes. Okay, because that's what first discovered, right? The chromatin or chromosome, those things, not the DNA. That's why based on the chromosome number, we gave the different names, reductional names. So what you have to, yes, whatever I wrote here, that is only for per cell. Okay, per cell. Even in the anaphase also, it's only for per cell. Okay, per cell, and that means a daughter cell. Okay, that's why. So these kind of names are given. Okay, now you have to remember two things. When the chromosome or DNA contents, when the DNA contents will getting doubled only in the S phase. Remember, only in the S phase of the interphase, the DNA content will get doubled. Okay, as you can see in here, from 2C to 4C happened, right? when it is getting reduced, okay, when chromosome number and DNA content will get reduced only in the anaphase stage, okay. So that may be anaphase 1 or anaphase 2. In the mitosis, it's a single anaphase. But where it will happen? Only in anaphase. So the changes in chromosome number or variation in chromosome number and DNA content only happen in what phases? Only happen in the anaphase stage or in the S phase. Okay, in S phase normally increase and in anaphase it will get reduced. Okay, so you have to remember this one, how it changes will happen, because they may ask question with related to the number of chromosomes, sorry, number of DNA content and the chromosome numbers also. Okay, is right. So just have to see the how many centromeres are there or how many chromatids are there. That's it. Okay. Any doubts, guys? If you have any doubts, just put it in the chat box. Otherwise, you can click on yes or no. Sorry, yes, so I can move on. Okay. If you click on yes, I think so you don't have any doubts. So I think I will move on. Okay then. So even in the YouTube also for the new people. Okay. So if you don't know anything, if you wanted to ask any question or anything, you can just put it in the chat box. So I will reply. Okay. Okay. This question, similar kind of question asked in NCERT also. Good. Okay. So we will move on then. So that we are starting a new chapter. Sorry, I took a lot of time. That means half an hour of time for explaining that. Okay. So morphology of flowering plants. In this particular chapter, we are going to study about different things. I will tell you. Okay. And for the new people who came to this class for the first time, my name is Subramanya and I'm taking your biology class. Okay. Sorry for introducing myself after half an hour. Okay. Leave it. Okay. I added the name here. That's why I introduced here. Okay. Then the first question came. Okay. Related to morphology of flowering plant. The question is simple. Angiosperms will follow. Okay, they are asking what angiosperms will follow. Is it haplontic life cycle? Is it diplontic life cycle? Is it haplodiplontic life cycle? Or no alternation of generation is found in here. Okay, I'm launching the poll and I'm giving only exact 1 minute of time because we see all these things in Plant Kingdom chapter and it's one of the easiest question if I remember properly, right? You have to see what kind of structures they are. Is it sporophyte or gametophyte? Then you will you can directly answer the question. I'm waiting from the YouTube. I know yes, some of you people forgot because the responses what I'm getting is telling everything. Okay. YouTube, I'm waiting. Correct. So last 10 seconds. I'm not giving any extra time. Okay. I'm ending the poll then. I'm sharing the results. 21% chosen option A, 40% B, 38% C, and 4% D. And in YouTube, everyone given option B only. Okay. There is a lot of competition is going on between B and C, right? And I know so many of you people are you should be little more quick, okay, because I already given 1 minute and 7 seconds of time. Only 50% some people are answered. Okay. Now tell me guys, what is the meaning of haplontic life cycle in a simple word? In haplontic life cycle, very, very simple word. If I want you guys to explain, then how can you say that in 1 or 2 word? 1 or 2 word. I just want 1 or 2 word. Okay. So reduction now. What reduction do? Exactly, right. Majority is and gametophyte is dominant. Haplontic life cycle means most of the life cycle will be haploid, right? Most of the life cycle is haploid. That means it will be in the gametophyte stage, right? So what is the other thing? The diplontic life cycle. Diplontic life cycle means most of the time it will be diploid. That means it will be in the sporophytic stage. Exactly, sporophytic stage. Sporophyte means spore producing structure, gametophyte means gamete producing structure, haploid haplontic means in between. right? So equal time will spend as the sporophyte and a gametophyte. And no alternation of generation means there is no sporophyte and gametophytic phase at all, right? No sporo and sporophytic and gametophytic phase. Gametophytic phase, exactly. Right. So here we are asking about angiosperms. And what's the example of haplodiploidic phase? Haplodiploidic phase. Can anybody tell me the example? If anyone remembered the example for haplodiploidic phase. So very. So this is also like you can answer in like seconds, right? Exactly. Bryophytum. You can take any bryophytum, right? And mostly the pteridophytes also. Exactly. Bryophyte and pteridophytes are the haplodiplonts. And I'll write down here only. The algae normally show, most of the algae will show the haplontic life cycle. That means gametophytic phase. And this bryophyte and pteridophytes will show the diplontic life cycle, right? So these are like half of their life will be in the sporophytic phase, half of them will be in the gametophytic phase. Exactly. Correct, Gopika. Exactly. But if I ask about diplontic life cycle, what is left in the plant kingdom then? Algae is over, bryophyte and pteridophyte is over. The only thing left is gymnosperm and angiosperm. What is that follow then? The angiosperm and gymnosperm, gymno and angio, will follow the diplontic life cycle. Okay, it will follow the diplontic life cycle. That means whatever you are watching, okay, whatever angiosperm or gymnosperm you encounter with, all of them are actually sporophytes, because the gametophytic stage is so tiny. Okay, it will happen within the pollen grain. Okay, and it will happen within the ovary. Okay, that much tiny the gametophytic phases. Okay, the major is sporophytic phase. So the correct answer is diplontic life cycle. So what's the example for no alternation of generation? No, in plants or in angiosperms, no alternation of generation you can't see, but you can correlate this with some of the fungi or the low level of bacteria and all those things. There is no alternation of generation. They only act as a single kind of diploidic kind structure. Okay, so we'll move on then. So what we are studying in this particular chapter? In this chapter, we are going to study about plant morphology. That means how the plant is and what's root definition and modification. And the next one is the stem. Stem means, so what's the stem and what are its modifications? Okay. Foliage means related to leaf. Okay, remember, whenever I'm using the term foliage or foliage, you have to think about the leaf, okay, or structures which is related to leaf. Okay, then inflorescence, which is related to the flower. Okay, how the flowers are arranged and how it's different uses or types. Okay, then fruits, fruits, so like dry fruits, and another one is the fleshy fruit. Okay, not wet fruit. So dry fruit and fleshy fruits, and how can we differentiate it? And this is kind of interesting. Okay, and seed, types of seeds. What are kinds of seeds are there? When we study all this character, we will go to 3 plant families. One is the Fabaceae, and another one is the Solanaceae, and the third one is the Liliaceae. Okay, so we will see some of the example, and we will a little bit go through about what are structures and how it will form and everything. Okay, this chapter is so interesting, guys. Okay, why it's interesting then? Okay, the major reason why it's interesting, because this will open up a different kind of languages. Okay, if you read already this particular chapter, you probably come across with more than 15 new words, right? More than 50, right? Some of the vegetables you normally see in here, we mentioned it under the fruit, right? Some of the seeds you say, this is actually a fruit. If I take an example, like cotton, we normally say like cotton ball or something like, but it's a fruit, right? But when you think about strawberry, strawberry will contain all the seeds above that, right? Seeds will be present above that, but those are not actually seeds. those are actually its fruits. Okay. What about the strawberry you are eating? What you are eating is just the thalamus. Thalamus is the base part of the flower. Okay. What you are eating is just the base part of the flower. What you are seeing is a seed. That's actually its fruit. Okay. So the changes which happened. Okay. And in the modification for the storage or for the different kinds, what are the changes happened in the plant, everything we will see. Okay. So once this chapter is over, so almost like a pretty morphology will be completed. So you can differentiate different parts of the plant and you can differentiate based on its functional. Okay. So if you have any doubt, I told you what you have to do. Just text it in the chat box. Okay. And whenever I complete a particular chapter in this morphology, try to look around you and try to check different plants and how modification happened and everything. When we are taking the offline classes, I normally take flowers or the plants to the class because then you can understand properly. But here I can't do that. But I'm bringing those things virtually. Okay, I'm just putting the photos and everything. You have to see here. But for experience, you have to go outside and you have to check it out for yourself only. Okay. Okay, then we will start then. The first thing is the morphology of the plant. Okay. So before we going to understand about the morphology of plant, the serious question is what is morphology? Okay. So we simply say it like morphology is the study of external character. Okay. But according to etymology, etymology means from where that word comes. Morphology is the scientific study of form and structure. Okay, how it is and how it's formed. We will study it in the morphology. Okay, so that's what we are doing it here with respect to the plant. Okay, a typical plant, if you take, you can differentiate into two different sectors or do different or major changes. One is the shoot system and another one is the root system. How can you say that then? The shoot is the one which is present above the earth and which is having a leaf, flower and everything. The root is the one which is present below the earth. Okay, so that's the like a broad differentiation. Okay, shoot means which is above the earth having leaf, and the root is the one which is below the earth. Okay, and shoot system normally consists of a leaf and a stem and a fruit and flower and everything, but the root will contain only the primary root and branched root, that means secondary root and root hairs and everything. Okay. And in this particular one, if you see closely, okay, we mentioned a node and internode. Okay, what's the meaning of a node and what's the meaning of internode then? If you take any plant, the leaves or the flowers are not coming randomly. Okay, these are coming at exactly certain points. Okay, the points where the leaves will arise, right? That particular point we call it as a node. Okay, exactly, node is the attachment site for the leaf, or we can see the vice versa also. Okay, the point where the leaf or the vegetative parts will arise, we call it as the node. Okay, you take any plant. Okay, you can take any plant. Here we are referring a single plant or flowering plant. You can take any plant from anywhere, wherever you can differentiate the nodes and internodes. Okay, in between two successive nodes, you can find a gap, right, where the leaves will not come. Okay, so that particular point we call it as the internode. Okay, probably you heard these terms for the first time, but I'm 100% sure that every one of you written these nodes and internodes or draw these nodes and internodes in somewhere. Okay, I will give a simple example. Okay, so probably most of you written the bamboo, right? Bamboo. So probably you written something like this, right? Bamboo plant, something like this, right? So in this particular bamboo plant, wherever you make some crevices like this, right? So this one is node and this one is also node. In between space will be internode. Really, nodes and internodes are in 7th. That's good then. I thought that some of you probably never heard of these things. Okay, no issue. We repeated it, right? Good thing. You can take care of, you can take an example of sugarcane also, right? Sugarcane also, you can see this kind of structure only. I think everyone understand what is the node and what is internode means. So you can only find node and internode in the shoot system, not in the root system. Okay, exactly. You can also find it in the coconut. Exactly, such right? Coconut, taraka tree, right? So you can only find nodes and internodes in the shoot system, not in the root system. Okay, so you have to remember that one. So we will move on then. So how we classified the plants then? We classified them based on different types. Okay, but we will consider based on lifespan. Lifespan means how much it will live on Earth or live on a planet. Okay, and the first ones are annuals. Okay, what's the meaning of annual? Annual means they complete their complete life cycle in a few weeks or in a few months, or you can say one growing season, or you can also say it in like 1 year. Okay, within 1 year. Okay, so why they are doing that? They are kind of lazy, you can say, or they are kind of smart, you can say that also. Okay, so reason is, if you are going through a summer season, then summer the water will not be there. Okay, then the sunlight will be too much. Then you have to store the nutrition, you have to store the water, you should be present near the water body, and you have to photosynthesize, and you have to reduce the risk of getting damaged with that kind of sunlight, right? It's too dangerous. It have to do a lot of work. But these plants are smart. Okay, what they will do, they will grow in the rainy season. Okay, they will produce flowers in rainy season or in the winter. Okay, and they will produce the fruit, they will produce the seeds, and they will die off when summer is coming. Okay, before summer is there, it's completely gone. Okay, the only thing left is the seeds. Seeds can tolerate what kind of temperature is given. Some seeds can tolerate up to the 90 degrees Celsius. Okay, so for seeds it not at all matters, right? So for escaping what I can say, escaping from the harder situation, these are become annuals or growing in a particular season. Okay, example maybe mustard. No, not like that, Chiran. Not like that. Okay, they can grow big, but they went for growing in a particular season to avoid the harsh conditions. Okay. And examples will be mustard, pea plant, or you can go for maize or rice plants. Okay, and like you can see, right, lot of herbs will be growing in the rainy season, but as soon as the winter and summer comes, everything already dried off, right? Those are the annuals. Okay, so you can go for finger millets, all of them. Okay, second on biennials. Okay, what's the meaning of biennials? They complete their life cycle in 2 years. Okay, so why they will do that? Okay, because in the first year they normally grow. Okay, and they will grow, they will store the energy, and they will do those things. And in the second year they will produce a more flower and fruits and seeds. Okay, that's why they need the 2 years. So we call it as the biennials. Okay, biennials, that means it needs more than 1 year but less than 2 years. Okay, so example is radish, turnip, and carrots. These are actually biennials, and these are belongs to the temperate regions. Okay, but when they are going in warmer region, that means near to the tropicals. Okay, so in the warmer regions, because of the more sunlight, okay, they will become what you can say annuals. Okay, they will act like annuals. That's why even though you are planting your carrot or radish in your farm, you will get a carrot or radish within 1 year, because they are act like an annual in warmer regions. Okay, if you plant the same thing when you go to like Europe or something in the upper hemisphere, okay, or in the like temperate regions, then they will act like the biennials. Okay, the turnip means, this is what turnip. Okay, turnip. So that's why I added this one picture only, because a lot of people having doubts in this one. Turnip, this one, it's almost equals to the nolkol. We normally see nolkol, right, in Navil Kosu, nolkol in Kannada. So that's also belongs to the same thing. This is not our native. none of these things are our natives, but this is not famous in our area. That's why we don't know about this. But all are almost similar because all of them are developed from a single plant only. Okay, mustard. Okay, mustard, wild mustard plant only. That's why almost everything is similar. Radish, sorry, on no no. Onion will take 2 years or 1 year. 2 years or 1 year. Onion. Onion, probably 2 years. Yeah, 1 year. I'm not sure about onion. Okay. Onion, wherever we are going, it's 1 year only, but I'm not about it's the actual biennial or annual. In our area, nobody will grow onion because due to this heavy rain, everything will be like, uh, everything will go. Nothing, we can't grow anything. Okay. That's why we don't know. Now it's different. Okay, now due to the selective breeding, we created such a plants. Those can be completed life cycle in very less time. Okay, so that's what happens, right? Exactly. So not only onion, radish, carrot, whatever you take, it will complete in few weeks, right? It's not at all taking few months. It's completing in a few weeks. Why it's happened? Because of our selective breeding, and we created some due to some genetic engineering and tissue culture breeding, we created a new varieties. Those can grow in a very less time. Okay. Exactly. So that's what we call it as hybrid. Yeah. So we'll move on then. Last one is the perennials. Okay. Perennials survive for several years. Okay. This plant usually flowers fruits every year, but they won't die every year. Okay. So example, mango, banana, guava, or any large plant, coconut, areca, everything, what you can see here. Okay. You may ask me something like, sir, banana plant will grow and give fruit in 1 year, some of them. Why can't we call it as like annual or biennial? Because probably if you've seen banana plant, you already know the answer. Even though the banana plant is give the fruit and everything is, it will die, still the base will remain like that only, right? The base is the plant. What it's giving is just a progeny. Okay. So almost all trees are perennial only because in 1 or 2 years, 2 years, it can't grow into a tree, right? So all the trees are perennial only, exact action. Okay. Herbs are differentiable. Okay. So these are related to annual, biennial, perennial, depends upon their lifespan. Okay. Do you have any doubt regarding this? If you have any doubt, just put it in the chat box. Okay. Otherwise, click on yes or put yes so I can understand. Even though not die, fruit giving efficiency will decrease. All those things will happen. Okay. But they will still there, right? Life is still there. Exactly. So that matters. So we'll move on then. Okay. If you have any doubt at any moment, just put it in the chat box. Okay. Especially in YouTube also. So next one is based on the nutrition. Okay. Based on nutrition, we differentiated plants into autotrophs and heterotrophs. When we are learning about the kingdom plant, we normally call it like photosynthesizing plants, right? Photosynthesizing organisms, we normally say. But I told you there, if you remember, not all plants are autotrophic, right? Not all plants are autotrophic. Some of them are there. Okay, those are not autotrophic. We call those kinds of plants are heterotroph. So what are the differentiation? The autotroph, that means they are photosynthesizing plant and they synthesize their own food utilizing water and CO2 along with the sunlight, right? They do the photosynthesis. They produce their own food in the form of glucose and store it in the form of starch or any other kind. So all green plants. Okay, simple. Green is there, that means chlorophyll is there, that means they are producing the food. Okay, but some of the plants are heterotrophs. That means they cannot synthesize their own food, so they are dependent upon other plants. Okay, as you can see here, leave this grass behind. Grass is autotroph. Okay, I'm not talking about this grass. This plant, if you can see here, okay, I'm not sure about this, but who having the maize plantation, okay, if you have a maize plantation, or almost throughout the Karnataka and also in the Maharashtra and Andhra Pradesh region, if you are going anywhere in the like shrubby kind of the forest, you can find these kind of flowers which is coming out of the this one, okay, land. If you try to investigate, you can't find any leaf or anything. You can only find colored inflorescence or colored flowers only. Okay, you can't find any green plants or green leaf or anything. So these are actually a parasitic kinds of plants. They normally feed on other plants. Okay, and they only produce their shoot to produce the spores and the seeds. Okay, because sorry, not spores, only the seeds, because the seeds should carry on, right? If it is produce the seeds inside the soil, it will be there only. That's why it will come out of the soil to produce the seeds. Okay, so just try to observe. Okay, so probably you may find one or two somewhere. I got it in like several part of the Karnataka actually. When I'm going to different places, I got it like two different parts, even in north, also in the south Karnataka. Okay. In labs for making new breeds, cells are observed to know lifespan. Uh, Not cells. If you want to do anything about why not spores, because spores are, I'm not talking about pollen grain or that kind of spores. Spores are produced in a lower organism, not in higher plants, right? That's Mohan. So for creating anything in the lab, before culturing a particular type of the plant, we already know all the background of the plant. Okay. So what's the lifespan, when it will produce the flowers, when it will produce the fruits, and how can we increase the content, everything. Okay. Then only go for the tissue culture and genetic engineering kind of the things. Okay. Those things you will learn in 12th. Those are like little easy to learn. Okay. Like to read it out, it's little easy, but for making the experiment is too tough. Okay. For breeding a particular type of the plant, for a simple breeding, it takes almost like 5 to 10 years. Okay, for simple breeding, I'm telling. So for testings, there are like too many steps are there. Okay, so but we already know all the things about the plants. Then we will proceed to the improving a particular character. For a single plant, we know you normally call hybrid, right? It is the sweat of scientists from 5, 10, or 15 years. Okay, you will learn that in 12th class. No tension now. Okay, so we'll come back to here. The plants on the basis of nutrition can be divided into autotroph and heterotroph. Autotroph produce their own food. Heterotrophs depend upon other organisms. Okay, so in heterotrophs, we are going through in detail. Okay, autotroph we already know. So heterotroph we are going in detail. So in that, the first one are parasites. Okay, you already know the meaning of parasite. Parasites means organisms which depend upon other organisms. For is all non-greens are heterotrophs. How can I put it? Not all greens are autotrophs. Okay, and is all non-greens naturally available, then it's heterotroph only. Okay, if it is non-green in naturally occurring, then it's heterotroph only. Okay, so this parasite normally dependent upon other plants. Okay, or other kinds of organism for their food. So we normally divide it into two kinds. One is the obligatory or total parasites, and partial or the semi-parasite. You already know the meaning of obligatory, so I'm first going to here only. Obligatory means they are completely dependent upon the plants for food and water. Remember, they are completely dependent. They are not at all doing any work. They are taking everything from the host. Okay, everything. So from the food, mineral to water, everything. Okay. So this can be again differentiated into stem and root parasite. Okay, the first is the total stem parasite. That means these are present on the other plant stem, and they will take the nutrition from the stem. Okay, the example is cuscuta. Probably most of you already seen a cuscuta, a yellow color vine or yellow color plant which is normally surround other plants. Okay, it is don't have any color because it don't have any chlorophyll or anything. Okay, that's a total stem parasite. We call it as cuscuta. Name is cuscuta only. Okay, so that's the stem parasite. What about root parasite? So the root parasite examples are Balanophora and Rafflesia. I don't think Balanophora name known to many people, but probably everyone know about the Rafflesia, right? What is the importance of Rafflesia? So can anybody tell me the importance? Or exactly, Rafflesia is the I just want from others also. Like, do anybody know exactly? Rafflesia is the largest flower. Rafflesia is the largest flower, exactly. So that Rafflesia is also a complete root parasite. Okay, this is also a complete root parasite. That means it will stuck to the other plant roots. From there, it will take nutrition and it will produce the flowers and everything. Okay, and this is also not green because it's normally present inside the soil only. Okay, so that's the total stem and total root parasite. Okay. So next one is the partial or semi-parasites. So these are depend upon plants for water and mineral only. They are not completely dependent upon everything. They depend for the cuscuta. This one is cuscuta. This one is cuscuta, and this all these are flowers of cuscuta. This is a small twine is there, right? This one is the cuscuta plant. Okay, it don't have any leaf or anything because it's not doing any photosynthesis. Okay. Remember, partial and semi-parasites are different. Partial and semi-parasites are dependent upon the other plant for water and mineral only, not for food. Remember, not for the food. That's why the partial stem parasites are normally green in color. Okay, they will green in color. The first one, partial stem parasites, that means they normally grow on other plant parts or other plant stems, and they will take the water and minerals. Example, Viscum and Loranthus. Okay, as you can see here. And there are some partial root parasites. So those will grow their roots, which attaching to the other plant roots, and they will take nutrition from their roots, but they are not dependent for the food. They are also green and they will also photosynthesize. Okay. The best example I can give for this is Santalum, which means sandalwood. Okay. Probably everyone's seen the sandalwood tree, or at least most of you already seen the sandalwood tree, right? The sandalwood tree will be big and it's having the green leaf, everything, it will produce flowers, fruit, everything, right? But for the first development, wait, Chiranta, that also come, okay? For the initial development, that particular sandalwood also will attach to some other plant roots and they will take nutrition from the other plant and they will grow. Okay, that's also a partial root parasite. Remember, the sandalwood, it's a partial root parasite. Okay. So this is the another one. Mohan, we will come back to that when we are discussing about flower. Okay, because otherwise our root will fall off. That's why. When I'm explaining there, you only understand why that happens. So probably I answered the question in the initial class, first or second class also. Okay. So partial root parasites also there, partial stem parasites are there. Both of them are green only, but these things are not producing or these are not taking food from the host. These are only taking water and mineral from the host. Okay. So any doubts regarding this parasite? Third slide, allowing no. This is not that one. This is just a fruit of the this one Viscum. Okay, this is just a fruit of Viscum. It's not anything else. Okay, we want to move on then, right? So next is the insectivorous plant. Now you got it, I think, right, Chirant? So what about insectivorous plants? Insectivorous plants are not parasites. Remember about that. Okay, insectivorous plants are not parasites. So, don't just ignore this pointers. Okay, so they grow in a soil which are normally deficient in nitrogen. Okay, when nitrogen is not in the soil, why nitrogen is not there? For nitrogen to be present in a soil, there should be some bacteria should be there which actually use the N2 or nitrogen gas and produce into some other like nitrate or nitrite or ammonia, and we leave it in the this one, okay, soil. But in some part of the area, so those kind of bacteria will not be present because it may be a logged, waterlogged area, or it is not suitable for those bacteria to grow. Okay, in that kind of areas, the nitrogen is very, very deficient. Okay, you already know nitrogen is very important in our body because that's what the constitute of amino acids, or you can take an example of DNA, everything, right? Those plants which is growing in those kind of areas come up with a new strategy to getting the nitrogen. Okay, what is that? They are not getting the nitrogen from the soil. That's why they are trapping the insect or other animals, and by digesting that, they are getting the nitrogen. Okay, like it's not getting from the soil, that's why it's getting from the outer sources. Okay, so these are also chlorophyllous plants only. Remember, don't think that this is also non-chlorophyllous. The insectivorous plants also chlorophyllous only. They also synthesize its own food, but remember, they only trap insect or animals for or as a source of nitrogen, not for the food. You have to differentiate that. Okay, it's what I'm telling. The insectivorous plants also chlorophyllous, and they will produce their own food, but they will trap or they will eat insect or other organisms only to get the nitrogen, not for the food. Okay, so all that's why what happens, all insectivorous plants, you can call them as the producers because they are producing the energy, and you can also call it as like secondary consumers because they are consuming the organisms. Don't mind about these things, you will learn that later on. Okay, the examples, Drosera will be this much small. Okay, mm, it don't have the nitrogen. No, Chirant. Mycorrhiza is only helpful for phosphate, not for the nitrogen. Okay. This is the Drosera. It will be 1 inch or 1.5 inch or 2 inch long only or large only. And Nepenthes, okay. So, and Venus flytrap, as you can see here, Nepenthes, Drosera, Nepenthes, and Venus flytraps. So this will normally go for the insect, but sometimes for their bad like this kind, like frog and everything is also gets stuck there. Okay, if you put your hands also, it will close the lid, but that's not strong enough to hold your this one fingers. Okay, but it's strong, but not that much strong. These are not this much gigantically big. These are very small. Okay, and the force is very less, so you can easily remove your hand. Okay, how they will digest, they will create some acids which will digest all the materials which is in the insect or in the animal body, then they will absorb that. Okay, obligatory insectivorous, no, there is no obligatory insectivorous because these kind of things never give the complete nutrition, or that much is not enough. It can't trap all the things, right? For completely getting everything from the this one, from the predatory nature, it should need a lot of prey, right? But that is not possible for the plants because plants will be placed in a single place. This is not moving, right? That's why obligatory insectivorous is not fine, but we can't use the obligator here. Okay, okay, then we'll move on. So one question is here, simple question is here. Partial parasites obtain their food from insects and animals, dead and decay materials, photosynthesis, host plant. Okay, I'm launching the poll, and you can answer in the poll, and who are in the YouTube live can answer in the YouTube live chat. Okay, so easy question, I think. Easy question, and like, how can I say, expected answer. Lankesh, check one more time what we discussed in there. Okay, you still have 30 seconds. Good. Okay, you guys have 15 seconds, last 15 seconds. I'm ending the poll in few seconds. Answer quickly. I'm ending the poll and I'm sharing the result. 18% A, 14% B, 22% C, and 46% D. And in YouTube, some people choose an option C and some option B. Okay, we already mentioned here, or we already told, this is not constitution to like select the things which most of the people are voted, right? This is different. We have to go for the correct answer. So we will see what happened here. We are asking about partial parasite and obtain their food from. Okay. So what is the partial parasite then? Partial parasite is the one which is growing on the other plants, and they will take water and mineral from the other plant, that means from the host. But what about the food? Okay, these partial parasites are green in color and they will produce their own food by the process of photosynthesis. So very less people are correct. Okay, so the correct answer is photosynthesis. Okay, so that's the correct answer. Exactly. Okay, so correct. So here, another one question. What are saprophytic plants? Okay, saprophytic plants are the one which normally grow on dead and decay materials. Okay, those plants are very rare, like some of the orchid species. And don't think that all these things are not present in Karnataka. All these kinds of plants are present in Karnataka. I already seen it several times. Okay, so don't think that it's a very rare thing. It's normally present, but we normally ignore that because these plants are very tiny. Okay. So saprophytic plants grow on dead and decay materials. Those are also normally non-green. Okay, non-green color will be there. That means brown, yellow, white color will be there. Okay, so I left it that because that's not that much important. That's why. Remember that also. Okay, so examples are not important. So first thinking, when we are going for a differentiation of a plant, we will start with a root. Okay, because in textbook it's given root. Okay, not any specific reason. So root, what is the meaning of roots? Roots are non-green plants, sorry, non-green part of the plant body, and they are present in underground. Okay, they are present in underground means below the soil, and they are positively geotrophic. What is the meaning of positively geotrophic? That means they will move towards the earth. Okay, they will move towards the earth. That means if you plant it in any way, the plant will always grow its roots towards the soil, okay, or towards the earth. Okay, so they are negatively phototropic. That means they are go away from the light. Okay, go away from light, Okay, and positively geotropic matlab go towards earth. And what's the meaning of hydrotropic? Positively hydrotropic. That means go towards water. Okay, remember roots are non-green. That means it do not contain green color, and these are present underground. Okay, that's not green color. That root, what you can see, it's a yellowish something color. Okay, yellowish soil color. So these are positively geotropic, that means go towards the earth, and these are negatively phototropic, that means go away from the light, and these are positively hydrotropic, that means go towards the water. Okay, and roots do not bear any buds. You normally see buds in the plant, right? But you can't find it in the root. And roots do not bear nodes and internodes also. So you can't find nodes and internodes in here. And roots have unicellular root hairs. If you observe the roots in microscope, you can find a hair-like structure on the root. Okay, so that is called root hair. Those are unicellular in nature. Okay, those are unicellular in nature. That's related to the root. So roots can be easily differentiated two types. One is a taproot and adventitious root. Okay, so if you take any seed, you probably observed a root-like structure which is coming from that one, right? Root and shoot, right? Root, and this one is the shoot. This particular root-like structure, we call it as the radical. Okay, we call it as the radical. Chemotropism, nutrients. But that's not always correct, right? Chemotropism, we normally mention it with the microorganisms, not with the plants or anything, roots, because everything is chemicals only, right? That's why. So, in a seed, you can find, in a germinating seed, you can find a radicle and a shoot. Okay. In some of the roots, the radicle will continue to grow and make a main branch, and from there, all the sub branches are divided. We call that kind of root as the taproot. The example is dicot root. Okay. Uh, Second one, adventitious root. So in adventitious root, what happens? Some plant, this radical will stop growing, but some other from other part, all the roots will start to come out. Okay, so that's what we call it as the adventitious root. Okay, we can normally found it in the grass species. Okay, no, not ficus tree, Chirant. Okay, Alamara is not the example for that is different adventitious. Okay, so this is different adventitious. Okay, so taproot means coming from the radical or containing the radical. Adventitious means apart from the radical. Okay, apart from the radical. So that's what it makes us confused. Okay, remember, adventitious root means any root which is coming out of other than the radical, we call it as the adventitious only. Okay, but what I'm referring here, this adventitious which related to the monocot plant. Okay, monocot plant. Probably you already know about that. Okay. I will see that one, Chirant. Okay. So we will next go through the parts of the root. Okay, how the different parts of the roots are arranged or everything. Okay, so we can do that in the next class. Okay, so I just launched the NPS. You can complete your NPS. If you have any doubt, you can ask now. Exactly. Adventitious root, you can call it like fibrous root. Sorry, I didn't mention that. Okay, fibrous root. Thank you, Akshay. Fibrous root. Okay, so both are same only. So if you have any doubt, you can ask now. Okay, previous page. I'm going to the previous page. Yes. YouTube, guys, do you have any doubt? Otherwise, who are new to here, we are available in Telegram, also in the WhatsApp. If you have any doubt, you can post it there also. Okay. And notes will be uploaded. Notes will be uploaded in the drive. You can access the drive links are in the group description. Okay. Plant Kingdom chapter, once we completed the cell division, we can, actually our regular plan is like we will complete the cell biology first, then we'll go to all the other things. Okay. But initially we will bind to complete the first 4 chapters. That's why we completed that. Then we went to the cell biology. I will check that, Chirant. Okay. No issue. Or you can do one more thing. You can take a screenshot and send it to me. That will be better for me. Okay. That's an easy way. For in YouTube, you guys still have like 10 minutes of time. You can ask any doubt or anything you want to discuss. Next, we will see about different modification. Okay, so some of the parts are not in the textbook. So different modification of taproot, adventitious root, and different parts of the root. Okay, all those things that may take one day. Once the root is completed, we can move on to the shoot. Okay. Okay then, if you guys don't have any doubt, then I will close the YouTube live. So if you have any doubt, you can ask me later in the Telegram also. Okay guys, so thank you.